Welcome to StudentPal. In this Grade 12 Biotechnology lesson, we are beginning Chapter 4, Applications of Recombinant DNA Technology. This chapter asks what happens after scientists can isolate, clone and express DNA. The answer extends from identification and agriculture to gene therapy, vaccines, antibodies, and therapeutic proteins. But an application is not complete when a construct is created. Identity, function, manufacturing quality, safety, ethical scope and appropriate oversight must all be demonstrated. By the end of this lesson, you will be able to connect each application to its molecular principle, evidence pathway, and limitations. Keep your notebook ready, pause whenever you need to study a diagram, and let us build the chapter step by step. Here is everything we are going to learn. We will begin with the principle of DNA fingerprinting and follow a sample through collection, extraction, amplification, fragment separation, allele detection and profile comparison. We will learn why multiple short tandem repeat loci improve discrimination and follow a SAM principle through collection is not a complete genome sequence. We will then distinguish a transformed cell from a stable, characterized transgenic organism and connect crop traits to agronomic testing, non-target effects, gene flow, and resistance management. Next, we will compare ex vivo and in vivo somatic gene therapy, examine recombinant vaccine development, and understand monoclonal antibody specificity. We will follow recombinant insulin and growth hormone from coding sequence to purified active product. Finally, we will organize the evidence ladder from construct to function, quality and safety, examine ethical boundaries, and practice full-mark CBSE answers. With that roadmap in mind, let us begin with the central question of the chapter. Imagine that a recombinant gene has been confirmed by sequencing. Has the application succeeded? Not yet. A crop construct must produce a stable trait without unacceptable agronomic or environmental effects. A therapeutic protein must have correct identity, folding, activity, purity, and manufacturing consistency. A gene therapy vector must reach the intended cells and be monitored for duration, specificity, and adverse effects. Every application therefore follows an evidence ladder: molecular identity, expression, biological function, product or phenotype performance, safety, and appropriate use. The higher the claim, the broader the evidence required. Throughout this lesson, ask two questions: what direct observation supports the claimed benefit, and what additional test addresses a realistic risk or alternative explanation? Those questions turn exciting biotechnology into responsible biotechnology. DNA fingerprinting, more precisely called DNA profiling in many contexts, compares inherited polymorphic regions. Short tandem repeats, or STRs, contain a short sequence repeated a variable number of times. Different repeat numbers create alleles of different fragment length. At one locus, many unrelated people may share an allele. When several independent loci are examined together, the combined profile becomes much more discriminating. A child normally inherits one allele at each autosomal locus from each biological parent, making STRs useful in kinship analysis as well as identification. A profile does not read the complete genome and does not by itself reveal every trait or disease risk. The scientific conclusion must reflect the loci tested, population assumptions, sample quality, and possibility of relatedness. In a diagram, show a locus with variable repeat number and connect repeat count to fragment length and detected allele. A DNA profile workflow begins with sample recognition, collection, labeling, preservation, and chain of custody documentation. DNA is extracted and its quality is assessed. Selected STR loci are amplified, commonly by multiplex PCR, using negative and positive controls. Amplified fragments are separated by size, and alleles are assigned by comparison with an allelic ladder and size standard. The resulting profile is compared with a reference or database according to defined interpretation criteria. Contamination can introduce extra alleles, degradation can remove larger fragments, and mixtures can produce signals from multiple contributors. Replicate analysis or an independent sample may strengthen confidence. Every transfer and analytical step must be documented because a scientifically correct laboratory method cannot repair uncertain sample identity. A full credit flowchart links sample, DNA, amplification, separation, allele calling, comparison, and limited conclusion. Profile interpretation separates observation from inference. The observation is a set of detected alleles at defined loci. If a reference contains an allele not explained by a clear single-source evidence profile, exclusion may be supported under the stated conditions. If the alleles are consistent, the profile is not automatically unique. The strength of support depends on allele frequencies, number of loci, relatedness assumptions, and analytical quality. Mixed profiles require contributor models and careful attention to peak balance, possible allele dropout, and artifacts. A laboratory control that shows contamination invalidates simple interpretation. Ethical handling also matters because genetic samples contain personal information and may reveal biological relationships. Avoid the exam error of saying a DNA profile match proves identity with absolute certainty. State that the tested profiles are consistent and that statistical and quality considerations determine the evidential strength. A transgenic organism carries introduced genetic material that is stably maintained and may be expressed. Detecting a marker in one treated cell does not yet establish a stable transgenic organism. The introduced sequence and its molecular structure must be confirmed. Expression should be measured at the relevant RNA or protein level, and the intended phenotype must be compared with suitable controls. In plants and animals, inheritance across generations may be tested. In microorganisms or cultured cells, stability during repeated growth may be important. Position effects, copy number or rearrangement, can influence expression, so independent transform lines should be compared. The final claim must match the evidence. DNA presence, expression, phenotype, and stable transmission are distinct levels. A strong answer follows delivery, selection, molecular confirmation, expression, phenotype, and inheritance, rather than treating the word transgenic as proof of every stage. Recombinant DNA technology can introduce traits related to pest resistance, nutrition, product quality, or environmental stress. Laboratory expression demonstrates a molecular event, but agricultural value requires field evidence. The engineered crop should be compared with an appropriate counterpart across environments for yield, trait stability, and agronomic characteristics. Safety assessment may examine introduced protein identity, exposure, potential allergenicity or toxicity, compositional features, and intended food or feed use. Environmental evaluation can consider gene flow, persistence, effects on non-target organisms, and changes in farming practice. Pest resistance also requires management because strong selection can favor resistant pest populations. Refuge or integrated strategies may slow that process. Avoid presenting an engineered crop as universally good or bad. Evaluate the specific trait, organism, exposure pathway, evidence, and management plan. Gene therapy aims to alter genetic function in target cells by adding a functional sequence, replacing or editing a sequence, or reducing harmful expression. The therapeutic design begins with the disease mechanism and the cell population that must be reached. Delivery may use viral-derived or non-viral systems, each with limits involving cargo, cell entry, persistence, immune response, and possible integration. Expression that is too low, too brief, or present in the wrong tissue may fail, while excessive or off-target activity may cause harm. Evidence progresses from construct identity to delivery, molecular correction, cellular function, clinical outcome, and long-term safety. Most established strategies focus on somatic cells, so changes affect the treated person rather than being intentionally inherited. Heritable interventions raise distinct scientific, ethical, and societal concerns. Gene therapy is a monitored medical intervention, not a general method for enhancement. In an ex vivo strategy, cells are removed from the patient, modified under controlled conditions, tested, and then returned. This can permit selection and characterization of the treated cells before administration, but collection, culture, conditioning, and successful engraftment may be demanding. In an in vivo strategy, the delivery system is administered directly so it reaches target cells inside the body. This can access tissues that cannot be removed and returned easily, but distribution, dose, immune response, and off-target exposure become especially important. Neither route is universally superior. Choose from cell accessibility, required duration, delivery efficiency, manufacturing feasibility and risk. Clinical monitoring tests benefit and adverse outcomes over an appropriate period. In a comparison answer, use parallel headings: location of modification, target cell control, delivery challenge, testing opportunity, and major limitation. Recombinant vaccines use selected antigens or genetic instructions rather than requiring the complete disease-causing organism. A gene encoding an antigen may be expressed in a suitable production host, and the purified antigen can become part of a subunit vaccine. Other platforms can deliver genetic instructions, so host cells transiently produce the antigen. Development begins with antigen selection and molecular identity, then examines expression, structure, and formulation. Immune studies measure responses such as specific antibodies and relevant cellular immunity, but an immune measurement is not identical to protection. Controlled studies evaluate dose, protection, and safety. Manufacturing must maintain purity, potency, stability, and consistency. A recombinant vaccine can reduce some risks associated with whole organism approaches, yet it still requires careful evidence. The complete pathway is antigen choice, production or delivery, immune evaluation, protection, safety, and quality control. A monoclonal antibody population is derived from one selected antibody-producing lineage or recombinant sequence and recognizes a defined epitope. Hybridoma methods combine an antibody-producing cell with an immortal partner, while recombinant technologies allow antibody genes to be isolated, modified, and expressed in suitable cells. Binding is only the first evidence level. Affinity describes binding strength, specificity asks whether the intended target is preferred, and cross-reactivity tests unwanted binding. Therapeutic action may depend on blocking a receptor, neutralizing a molecule, delivering a payload, or recruiting immune functions. Diagnostic use requires reliable distinction between positive and negative samples. Product purity, aggregation, stability, and batch consistency also matter. Do not state that monoclonal means automatically safe or perfectly specific. The useful claim is established through molecular characterization, functional testing, and application-specific evidence. Recombinant human insulin illustrates how gene cloning becomes a manufactured medicine. Because bacteria cannot remove human introns, an intron-free coding sequence or designed construct is used. The sequence is placed in an expression system compatible with the production host. Depending on the strategy, insulin chains or a precursor are produced, recovered, and processed to create the correct active hormone. Purification removes host cell proteins, nucleic acids, endotoxins, and other process-related impurities. Sequence and mass analysis support molecular identity. Structural and biochemical tests assess correct processing, while a potency assay measures biological activity. Sterility, and batch consistency are essential for a medicine. A bacterial colony containing insulin DNA proves only an early molecular stage. The therapeutic product is supported by a chain of expression, processing, purification, functional, and quality evidence. Recombinant human growth hormone is produced by expressing the appropriate human coding sequence in a controlled host system and purifying the resulting protein. Molecular identity, purity, folding, and biological activity must be demonstrated, and manufacturing must control contaminants and variability. These achievements explain how the product can be made. They do not decide who should receive it. Appropriate use depends on a medically established indication, individual assessment, dose, and ongoing monitoring for benefit and adverse effects. Unsupervised use for performance or appearance is not justified by recombinant origin and can cause harm. This distinction is important in biotechnology. Technical production, product quality, and clinical appropriateness are separate evidence domains. In an exam answer, describe production and validation scientifically, then state that diagnosis and treatment decisions belong within qualified medical care and regulatory oversight. Across all applications, use one evidence ladder. First, confirm the construct or genetic profile at the molecular level. Next, test expression, or the immediate biological mechanism. Then measure function, a phenotype, protective response, binding effect, or therapeutic activity. Product applications add purity, potency, stability, and manufacturing consistency. Organismal and clinical applications add performance, adverse effects, and long-term monitoring. Environmental uses add exposure, persistence, gene flow, and non-target effects. Ethical responsibility includes informed consent, genetic privacy, animal welfare, fair access, ownership and communication that does not exaggerate certainty. Controls and independent methods reduce alternative explanations, while oversight matches the scale of risk. Biotechnology becomes responsible when its claims are limited to the evidence and its use is governed by safety, ethics, and accountability. For a 3 or 5 mark CBSE answer, avoid listing applications without explaining them. Begin with the molecular principle. For DNA profiling, write STR variation, amplification, separation, allele comparison, and interpretation limits. For a transgenic organism, write delivery, selection, molecular conformation, expression, phenotype, and stability or inheritance. For gene therapy, identify the target cells, delivery route, intended genetic change, and safety monitoring. For a recombinant vaccine or protein, connect coding sequence, expression host, purification, functional testing, and quality control. For monoclonal antibodies, add specificity, affinity, cross-reactivity, and intended function. A labeled flowchart can organize the process, while a short final sentence should state a realistic limitation, risk control measure, or ethical boundary. Full marks come from causal order and evidence, not from promotional language. Close your notes and rebuild the chapter. DNA profiling compares multiple inherited STR loci through a controlled sample-to-profile workflow and supports a limited statistical interpretation. A transgenic organism requires DNA confirmation, expression, phenotype, and stability or inheritance evidence. Agricultural applications move from laboratory trait to field performance, safety, and resistance management. Gene therapy must reach target cells, alter function appropriately, and be monitored for persistence and adverse effects. Recombinant vaccines connect antigen design to immune response, protection, safety, and manufacturing quality. Monoclonal antibodies require specificity and function testing. Recombinant insulin and growth hormone require correct expression, processing, purity, and potency, followed by appropriate clinical use. If you can move from molecular principle to function, quality, safety, limitation, and ethical scope for every example, you understand recombinant DNA applications as evidence-based biotechnology.